we are going to do a little lab today, or at least get the lab set up. We'll do the lab live during anatomy this afternoon. So I am doing the Dialysis and Diffusion Lab, which is posted on Canvas. Yes, my head is cut off, but that's so you can see everything else. So we have prepared 2 pieces of dialysis tubing. I've tied off one end with a rubber band, and I stuck a clean dropper in there just to hold it open. The most time-consuming part of the lab is getting this open so that we can put stuff in it. I have prepared 2 of them. I have prepared a starch solution, and I have prepared a sugar solution. In class today, we will be putting those into the tube, or the dialysis tubing, and then we will be placing them in a water bath. So I have one for the starch tubing, and I have a water bath for the sugar tubing. We're going to set this up at the beginning, and then we're going to let it sit here for 20 minutes, and then we're going to test to see how the starch and the sugar, how or if they're able to move across our model cell membrane, the semi-permeable dialysis tubing. So we're set back up again. We're ready to carry out our experiment. We're going to start out first of all with the starch solution. I'm going to put a little bit into this graduated cylinder. All right, it makes it easier to pour it into the dialysis tube, which is going to serve as our model of the cell membrane. Pour it slowly, because the stuff wants to stick to itself. All right, and then we're going to close off the end, give it a twist, give it a fold. You would think that a rubber band would be more difficult to do this with. But actually, when we've tried to tie this off with string, it's been harder to juggle and not spill. All right, I'm going to go over here now, and I'm going to rinse to make sure that I have no starch solution on the outside. I'm going to put this in the bath labeled starch. Set that right there. You can see it's labeled for starch. I'm going to move my starch solution off over here to the side. All right, now we're going to repeat the process for sugar solution in a different graduated cylinder, because we don't want any contamination. That would make a mess. All right, again, we pour slowly. Let it separate itself out. All right, twist, bend, wrap. All right, I'm going to go ahead and wrap this up and then rinse it. I'll put it in the water bath that's labeled for our sugar solution, which we have right here. And then we'll come back in 20 minutes or so, and we will see what we come up with.
Okay, we've let our time run for our dialysis tubing lab. It's time to label our well plates. We have a plate that I'm going to label starch. And according to the lab directions, we have well 1, well 2, well 3, and well 4. All right, so we're going to start with the starch solution. I'm going to take a little bit of water. Here. All right, so So we're going to take a little bit of water in the pipette. We're going to put that in well 1. And we're going to put that in well 2. We're going to take the little cell out and take the rubber band off. This is the other advantage to doing this with a rubber band, is that it will come right off. Most of the time. You can get creative and break the rubber band. Not optimal, but sometimes that happens. All right, so now I've got my tube. I'm going to open it up, and we're going to test the solution inside to confirm that we do in fact have starch inside. All right. And then this whole thing we are just going to dispose of, so I can just throw it all back in there. All right, the test. We're going to test for the presence of glucose because we want to make sure that we don't have any contamination or see if anything strange is going on. I have these glucose test strips. I'm going to take the little yellow pad. I'm going to put that in well 1. And then we're going to let that sit and see if it turns green. And I'm going to put it in well 3. So well 1, we're testing the water bath. Well 3, we are testing the solution in the little cell. Now the standard starch test involves iodine. So we're going to use this. We're going to put a couple of drops into our well plate, and then we're going to put a couple of drops. Let's put 3 drops in each one so we really see what's going on. All right. So you can see right away we have a color change from the solution in the dialysis tubing. We see no color change in the water that the tube was in. So this test tells us that we appear to have no glucose in either situation, as matched to the color on the pouch. But we do have starch in the tube, but not out of the tube, telling us that the starch does not travel across the membrane, which makes sense. Starch is a very large molecule. All right, we're going to repeat the tests with the glucose solution. All right, so we've seen this. I'm going to take my 2 test strips and move them up. I'm going to move this out of the way. I'm going to bring in my second well plate. I'm going to remind myself this one is for sugar. And I'm going to number 1, number 2, number 3, and number 4. Repeat the process with a new, fresh, clean pipette. We're going to take some water again from the solution. We're going to put it in well 1. We're going to put it in well 2. All right. Well 1 gets the glucose test strip. Well 2 gets our iodine. And again, we see no evidence of color change at this point. So now we're ready to open the sugar cell. See, that's how it's supposed to come off. All right, so we have our sugar cell. We're going to go in here. We're going to go in with our tube into the tubing, take some solution out. I'm going to fill up one well and the second well, and then we're done. And this is all going to get rinsed away. All right, glucose test strip number 3, or for well 3. All right, so, whoops. Three drops of iodine. All right. So, as we look at this, we find that, whoops, put it on the marker. All right. So we find that in this case, as we let it sit, we are seeing a color change on the glucose test strip. And we are seeing that we're getting that same color change just a little bit behind. So we're confirming here that there was glucose in the solution that was in the tubing and that it did get out. Now the interesting thing about this is that I set all this up to video for the anatomy lab, which actually happened yesterday. And we went through and we did the lab, we let it sit for the 20 minutes. At the end of 20 minutes, we found no glucose in the water outside of the tubing. But the computer that I was using to film yesterday, the battery had died, so I set it all up again to come back and film this part now about 20 hours later. And you can see that over 20 hours, some glucose does manage to get out of the dialysis tubing. How does glucose move in and out of the semi-permeable membrane of the cell? It moves through through facilitated diffusion through transport proteins. But if you let it sit long enough, some glucose will move through the dialysis tubing. Now we've got this. We've let both of these sit now for the 2 or 3 minutes that's required. We see very similar colors. They both look like they're at about the same, probably in this 300 range. So after 20 hours, we see that glucose did get out. When we let it sit for the 20 minutes in class, our results looked more like this. All right, we're going to record the data, our results, our observations. So with the sugar cell, that's our dialysis tubing. For the dialysis tubing containing sugar, we tested for glucose in the beaker. Here's the catch. "20 minutes, yellow, negative. 20 hours, green, positive." That was our weird scenario. So we hit it right out of the gate, be honest, up front. In the sugar cell, we tested starch in the beaker. That nice gold and iodine color, negative result. In this, we tested the starch tubing, glucose in the beaker. It was yellow, negative result. The starch cell, we tested for starch in the beaker. That nice gold iodine color, negative result. All right. So we got the same results, 20 minutes when I did it live in class, 20 hours when I recorded it for you. So the only positive result, we found glucose in the beaker water after 20 hours in our baths. What about now in the tubing? In the sugar cell, did we find glucose? Both times, yes, it was there, positive result. In the sugar cell, did we find starch? No, it stayed that nice gold iodine color. In the starch cell, did we find glucose? No, the glucose strip stayed yellow, negative result. In the starch cell, did we find starch? In the tubing, it was turning dark, positive result. If you have a really starchy solution and you drop a little iodine in there, it'll go to a dark blue, almost black. Thanks for watching. Hope this helps. Remember, we've got substances that are moving by diffusion through dialysis tubing, very similar to the process in our cell membranes.